Entonces, voy a presentar a Guillermo. Me da muchísimo gusto presentar al doctor Guillermo Oliver. Él, como algunos de ustedes saben, también fue parte de nuestro instituto, porque estudió aquí su maestría en el laboratorio del doctor Bolívar, y justamente en la etapa en que se inauguró el entonces ENGEVI, él fue de los que se mudó de biomédicas para acá. Entonces, pues lo conocemos muchos de los que trabajamos desde hace mucho aquí. Es originario de Uruguay y después de su etapa en México hizo su doctorado en la Universidad de Uruguay. Posteriormente hizo estancias en institutos importantes en varios países, como el Max Planck en Alemania, con el doctor Peter Gross. Después, en los 90, se estableció por un tiempo largo en St. Jude Children's Research Hospital en Memphis, en Tennessee, en donde ha hecho un trabajo muy destacado, particularmente en el tema del desarrollo linfático. Él, por ejemplo, fue el que descubrió el gen PROX1, que es como el master gene del sistema linfático. Su trabajo también reveló la relación entre los defectos del sistema linfático con la obesidad y otros desórdenes metabólicos. Bueno, tiene más de 100 publicaciones, muchos reconocimientos y muchas de sus publicaciones en revistas del más alto impacto. Y ha recibido reconocimientos y actualmente es director del Center for Vascular and Developmental Biology, Robert Lurie, en Northwestern University en Chicago. Y bueno, pues te agradecemos, Guillermo, doblemente por hacer este esfuerzo de hablar a estas horas, que es muy tarde ya para ti, y triple por buscar alternativas ante las dificultades que se te están presentando. Muchas gracias, te escuchamos. ¿Puedes compartir?
Sí, ¿me ven ustedes o no?
Sí.
Sí. Bueno, primero, sin duda es un gusto y les agradezco que se hayan acordado de mí. Sin duda, los recuerdos que yo tengo de mi época ahí y los amigos que mantengo son lindísimos. Así que es un gusto. Espero nada más que el internet funcione, porque estoy usando de mi teléfono, dado que acá el internet del hotel se cortó. Así que esperemos lo mejor. Si se corta, pero bueno, voy a hablar en español nada más al principio y luego voy a pasar al inglés. Lamento, pero tengo todos los slides en inglés. Así que, bueno, voy a compartir mi PowerPoint. Déjenme ver un poquito. Desktop 1. Share. Allow Zoom.
Está intentando compartir.
Ok, veamos. ¿Ustedes ven el PowerPoint? ¿Me oyen?
Todavía no.
No lo vemos.
¿Ven el PowerPoint?
No.
¿Qué es lo que ven?
A ti.
Te vemos a ti acá. No, bueno, en la que deberíamos ver el PowerPoint, seguimos viendo pues el auditorio. Pero no.
Ok. No entiendo muy bien por qué.
¿Sí te da la opción de compartir pantalla?
Sí, y según esto ya lo había hecho, pero...
Te voy a hacer... Omar, ¿podemos hacer algo?
Es que yo hice el compartir el... No entiendo por qué no...
¿No le puedes pasar Wi-Fi a una computadora desde tu teléfono? ¿Es lo que estás haciendo? Pero de su compu, de su teléfono a la computadora.
Share screen, hold on. Whiteboard 5.4, we are playing. Listo. Tengo que ver cuál de todas estas. A ver, voy a ir a...
Sí.
¿Todavía no? ¿Nada?
No lo vemos.
A ver, voy a seguir intentando. Allow to share your screen. Open system preferences. No entiendo por qué. ¿Por qué? Digo, no he tenido, lo he hecho 1000 veces y nunca he tenido problemas. No entiendo cuál es el problema ahora. ¿Por qué no me deja? Espérenme un poquito, capaz que alguien me ayuda. Esperen porque, estén conmigo un segundo, ¿ok? Sorry. Maybe you can help me. I'm trying to share the screen with them, and for whatever reason, still they see me. But I don't know what I'm doing wrong.
You in Zoom?
I'm in Zoom.
Okay. There should be a screen share button in Zoom.
Right, normally there is.
Can you open the full size panel?
This?
Just change back to the full size view.
This?
Sure.
Oh, which? This?
Yeah. No.
This is what they are, but.
Here.
Yeah.
Go on.
What?
Okay. And you're trying to share PowerPoint?
Yes.
Open system preferences. Allow screen share. Will not be able to record the contents of your
No, I
screen
don't
until
want
it is
to
quit.
record it, so.
That means share.
Oh, okay, that's great.
So you have to quit and reopen Zoom. Is that okay?
Well, we need to reopen Zoom? Yes.
¿Qué pasó? Creo que se tiene que volver a conectar. Sí, creo que tienen que reiniciar. Bueno, si alguien me sugiere que me mande la presentación, pero sí, lo que pasa es que no tengo comunicación con él, pero a ver si no, sí. Sí, ahorita en este momento no tengo comunicación con él.
Me preguntan dónde se pueden conectar para verlo. Susana López. En el mail que tú mandaste, ¿no? Ahí estaba la dirección.
Sí, a ver, lo voy a poner. Espérame.
O mándalo al WhatsApp del departamento.
Okay, thank you. Yeah, I appreciate it. Don't close me here. Okay, then. ¿La ven?
Sí, ya vamos a avanzar.
Déjenme sentar. Oh my God, what a... Okay. Okay.
¿Quieres ponlo en modo de pantalla
Sí.
completa?
Sí, voy a tratar de esperarme un poquito. Where are you?
Ok.
Okay. Ahora sí.
Ujú.
Okay, bueno. Sorry for the inconvenience. Let's start again. So what I want to do today is I give you a little bit of an overview of what I have been doing in the last 30 years or so. So hopefully you will enjoy as much as I am enjoying the type of research I do. But first, let me just give you a little bit of very briefly background about what I did and how I ended where I am. Ideally, if you have students there, it may give them an idea sometimes that how you can find different paths in different ways. So yes, as Hilda said, I trained in Uruguay. I got a biological science degree, and all my training was basically analyzing meiosis in crickets. So I was squishing testes of crickets and looking chromosomes on the microscope, and that was my training pretty much. And for different reasons, which is a little bit long to explain, I went to Mexico, and I was lucky enough to end initially at Biomedicas, where was shortly after Paco returned from San Francisco. And the picture there, you might recognize some individuals there, including myself. And it was a great time. I really learned a lot. It was the seminars we have. I was doing a master's degree. It was remarkable. I really learned a lot. And then from there, one day, Paco told us we were moving to Cuernavaca. And remember very well, we went to this mountain. There was nothing there. And he said, here is going to be the institute. And that's what it was. And that's where you guys are right now. And as I told you, my training was not in molecular biology. It was mostly in just cricket. So it was not easy for me to learn even how to use a pipette. I remember I was looking at Mario over my shoulder at the beginning. I was ashamed to ask how to use a Gilson pipette. And but anyhow, I more or less managed, and this is the type of things that you see the papers I published together. At the time I was in Paco's lab. Mostly I collaborated a lot with Fernando Valle. But I learned a lot of molecular biology. The best molecular biology I ever learned, it was there, and I'm very happy of what I learned there. And but I somehow always knew that I wanted to do developmental biology. The only problem, I didn't know how. And this diagram that is here, it represents a little bit what I always think, and I still think today. The critical step in your life is gastrulation. And as it says here, it's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. And these represent my thinking. Developmental biology is critical. If we don't have organogenesis, nothing else matters. The only problem was I wasn't sure how to study what I wanted to study. Until one day in the library at the institute there were these two papers back to back. One in Drosophila from Walter Goering's group in Switzerland, the other in mice in Xenopus, from Edith de Robertis' lab. And describing the first cloning of homeobox-containing genes. These are transcription factors that were the first kind of markers that allow us to distinguish genes which have something to do with developmental biology. And somehow knew this was the possibility for me to enter into what I wanted to do, developmental biology. So it took me a while. I wrote to both of them that I wanted to move with them. Obviously, I didn't have a big CV, but eventually through persistence, Eddie took me and I moved to UCLA with Beatriz, my wife, which is the other present I got in Mexico. And many of you know them, you know her as well. And this is the group where I was at UCLA, and this is the kind of the first review we wrote in Scientific America at that time. And from there, was a little bit of different adventures we have. We went back to Uruguay there was an opportunity to go back. It was not easy. Was to adjust after a decade abroad. And then we decided we wanted to leave. We were very close to go back to Mexico, but for different circumstances, it didn't happen. So we ended in Germany at the Max Planck Institute, where there was a big group working in other type of homeobox-containing gene called PAX genes. And again, we learned a lot. It was a great time, complicated. We have 2 young kids. We didn't speak German, but we survived for 5 years. And it was at that time that this adventure I'm going to tell you today started with this gene called, and now we call it PROX1. As I told you, I was very interested in genes, in homeobox genes, because somehow they labeled development. And one day there was a paper in a gene in Drosophila called Prospero. And I was very interested in this gene because, first of all, it was a very diverged homeobox containing gene. And that was good because meaning that probably it didn't have compensation by other family members. But the other thing, this gene in Drosophila was extremely important for the differentiation of certain type of neuron called neuroblasts and determine who is going to be a mother cell and who is going to be the daughter cell. So at that time, it's not like now that it's so easy to clone a gene. At that time, you needed to do radioactive screening libraries, etc., etc. It took us a while with Beatriz, my wife, to screen using the Drosophila probe, but eventually we ended with this gene, which was the kind of mouse homologue of Prospero, which we call it PROX1. And as you see in this cover here, it was beautifully expressed in mostly the entire central nervous system, beside other organs like the liver, the pancreas, and the heart. But I was very interested in the CNS at that time. Just to make the story short, during that time, I also generated a knockout construct for this gene. Basically, at the time I did that, I included a reporter, beta-gal, that allow us to follow the expression of this gene. And what you see here are X-gal staining beta-gal expression of this PROX1 gene during mouse embryonic development. As you can see here in blue, is very nicely expressed along the spinal cord, but it's also expressed, as I said, in the liver, in the eye, etc., etc. When we did the knockout, just to make the story short, it didn't have any phenotype in the CNS. The CNS was perfectly normal. But as I said, the PROX was expressed in a variety of tissues. We were able to publish a number of papers describing the role of PROX1 in the lens, in the retina, in the brain, in hepatocytes, in the liver, in muscle cells, and even in tumors. But lymphatics were not still in the picture at that time. Until one thing we noticed was, this was with Jeff Weigel, my first postdoc at the time. There was a very intriguing expression. I don't know if you can see here on the side. There are a few cells on the side of the embryo that gets increasing numbers as development proceeds that we didn't know what they were. We thought initially these were a novel class of neural crest cells that we later figured out this was incorrect. It was thanks to a colleague at St. Jude. Well, I must say, after we were in Göttingen, we decided it was time to move, and that's how we got an offer to have to start our independent labs at St. Jude Children's Research Hospital in Memphis, which is where we started. I started with this project, and we stayed at St. Jude for 20 years almost. So bottom line, we didn't know what these cells were. There were no neural crests until someone, a colleague of mine at St. Jude, said, "What about lymphatics?" And I was a little bit puzzled because, to be honest, I didn't know anything about lymphatics, and even the name sounded boring to me. So therefore, what I decided was to start reading about lymphatics. And but as we moved forward into this became quite interesting because turned out that indeed those were lymphatics. And bottom line, to summarize many years of work, what you see on the left, first we discovered that PROX1 was the first lymphatic specific marker. And that's what you see here is the skin stained with antibodies. In green is lymphatic vessels, and in blue are blood vessels. And the mutant embryos die at around E14.5. That means mid-gestation. And you see in panel A, the dermis is really swollen, and that's called edema. It's a typical feature of lymphatic malfunction. And later on, you see the lymphatics spread all over the embryo. But this was not only a marker, it was the first mouse model completely devoid of lymphatic endothelial cells. And that was something quite exciting at that moment. We published this in Cell in 1999. And just let me probably, you are wondering I will give you a little bit of background on lymphatics and why it was not much known at that time. As you know, we have two major vascular networks. The one that you're probably familiar, which is the blood vascular system, which is lined by endothelial cells. And then you have the other one, the one that is going to be the focus of my talk, which is the lymphatic vascular network, which is also lined by endothelial cells. But at that time, when we started, there were no markers for lymphatics. And that's probably one of the reasons why this field didn't progress, because at the naked eye, it's not easy to distinguish one vascular network from the other. Lymphatics run almost everywhere along the body and then eventually connect with the blood circulation at the level of the thoracic duct here. And basically, they bring back all the fluids that they absorb from our body back to the blood circulation through that lymphovenous valve. Lymphatics come in two different flavors, if you like. You have a smaller lymphatic capillaries, which are here depicted in panel B. And then you have larger collecting lymphatic vessels in panel C, which are surrounded by smooth muscle cells, and they have lymphatic valves, which is what pulls the lymph to flow along the body. And they eventually connect with lymph nodes, which are, you see it here. And eventually, as I said, through all that, they all go back to the blood circulation. Then we have a type of specialized lymphatics, particularly in the mesenteric region of the gut, which are inside the villi of the gut, which you see in panel E. And these are called lacteals. And all our lipids are basically packed in small particles called chylomicrons. And those are absorbed into the blood circulation, into our body through these lacteals. They penetrate through the lymphatics, the lacteals, and eventually they enter into the blood circulation, and that's the way we absorb the lipids, and that's a major function of lymphatics. So, but when we started to work on this, when we found that PROX was expressing lymphatic, pretty much there was nothing about lymphatics. And this is more or less represent what is our knowledge on lymphatics. There was a description which initially people think is the initial description of lymphatics in the 1600s by Gaspar Azelli, that they call it Y veins. But then we move all the way to the 1900s, in which a remarkable woman called Florence Sabin in the United States, using ink injections in pig embryos, she described basically a developmental model in which she says the lymphatics are venous derived. But then we need to wait 100 years, and where there is another paper, basically by a group in Finland that describe a receptor called vascular receptor 3, which they propose that is somehow expressed in lymphatics, but there is not much on that paper either. And then it comes our paper in 1999 that for first time identified a lymphatic specific marker and the first specific mouse model completely devoid of lymphatic endothelial cells. So bottom line, this field was completely ignored for a long, long time, and it was considered to be a boring system in many ways. We read a lot because, as I said, there was not much known about this network. And this is what I was telling you. There was this paper from this Kari Little group in Finland that report that this receptor VEGFR3 was somehow expressed in lymphatics, and the ligand called VEGF-C somehow also was involved in that process, but there was not much than that at that time. And this is because it's important for what I'm going to tell you later. So remember, keep in mind, in the process of lymphatic formation, there are these major players, which is vascular receptor 3, and its ligand, basically vascular VEGF-C, which is listed here. So obviously we were extremely intrigued. Now, how is PROX regulating because the process of lymphatic formation? Because as I said, when you don't have PROX, there are no lymphatics and there are no lymphatic endothelial cells. Obviously, the question is, how is PROX controlling the process of lymphatic formation? And a very detailed analysis of the process of lymphatic formation show us that in mouse seems to start around in 9.5 in the cardinal vein, in the anterior cardinal vein. What you see here is an anterior section and a transverse section of the anterior cardinal vein in the mouse, where you see just the surface of the vein, called cardinal vein, and what you see in red is immunostaining for PROX1, which is expressing what we are called today lymphatic progenitors. And we are pretty certain that this is the stage when the process of lymphatic vasculature formation initiates by the expression of PROX1 in a population of venous endothelial cells that become lymphatic progenitors. And obviously, the reason why when you delete PROX, you don't have lymphatics, because you arrest this process very early, meaning you don't have progenitors, therefore you don't have lymphatic vasculature in the entire embryo. So that was quite a good start, but obviously still we didn't know a lot about this process. So through the work at that time, we somehow proposed that this formation of lymphatics consists of 4 stages. The one I just told you, which is the expression of PROX in lymphatic progenitors. Eventually, these cells, we saw that they start to leave or bud the cardinal vein in a very precise manner. And we know that later on, these cells get together to form what are called lymph sacs, which are distributed along the anterior-posterior axis of the embryo. And by sprouting and proliferation, eventually they form the entire lymphatic network, as you see in panel D. And this is what I told you. This is the basis of what we have at that time, was the ink injections in pig performed by Florence Sabin that somehow really nicely mimic what we described 100 years later in our mice. And this is basically the basis that we use for our analysis. And this is later on another postdoc in my lab, Satish Srinivasan, decided to really validate because what Sabin proposed at that time was that the lymphatics were venous derived, which is more or less what we saw, that PROX expression starts in the cardinal vein. And what Satish did was to do a detailed lineage tracing to see indeed what was the origin of lymphatics. And I cannot go in detail, I don't have time. But pretty much what he was able to show that is to validate Florence Sabin's proposal, indicated that by this lineage tracing that most lymphatics in the mammalian embryo are venous-derived. In the last 4 or 5 years, there has been other papers that suggest that for certain tissues, there may be a small contribution of lymphatic progenitors for other sources. Many of these are still controversial and need to be validated at the functional level. But bottom line, the majority of the lymphatics in the mammalian embryo are venous-derived. The other important data we found later on, because as procs are embryonic lethal, we decided to eventually generate a conditional mutant that by crossing with Cree and tamoxifen injections allow us to delete procs wherever we want, whenever we want it. And that's exactly what this postdoc, Nicole Johnson, did in my lab. She deleted at different stages, embryonically, postnatally, and in adult. And the results were consistent because every time she deleted procs, what she saw was that these lymphatic endothelial cells reverted backwards to be what was the original fate, meaning to be a venous endothelial cells. And this is what you see in this panel here. We use a lot of markers of venous and lymphatic endothelial cells. And when you delete procs, they start to express again this venous fate. So what we proposed at that time, and now is completely certain, is that lymphatic endothelial cell identity is a plastic fate and require constant procs expression for its maintenance. But this also brought a very interesting question. Why you need plasticity? Why you need procs expression through life? And we know that not only lymphatic endothelial cells are plastic, but also the other two types of endothelial cells, arterial and venous, are also plastic and are regulated by mainly one gene. Arterial become arterial through the expression of Notch. Then venous become venous endothelial cells because CUPTF2 represses Notch and they become venous. And then now we show that you add procs on top of that and they become lymphatic endothelial cells. The question was, is this plasticity something that really happens in real life? And we still don't have a lot of answers for that. But one thought we have at the time, and we haven't published this, that maybe this plasticity is an advantage for certain settings in which you need a rapid supply of blood. And for example, we thought that could be true in tumors, in which a tumor needs a rapid supply of blood. And if it only happens through angiogenesis, that will be very slow. Instead, you have the nearby lymphatics. What if you just shut off procs and they become a functional blood vessel? We generated a mouse model of a highly angiogenic tumor, which is an angiosarcoma. And by lineage tracing analysis, we demonstrated that indeed that was the case because the blood vessel or the vasculature surrounding that tumor is highly hybrid and is a mixture of blood and lymphatic endothelial cell fate. And was very rewarding that just a month or so ago, there was a paper in Nature in zebrafish in which they showed that when you cut the caudal fin of the fish, which, as you know, the fish is highly regenerative, the new blood vessels that enter into the caudal vein originate from a de-differentiating lymphatic vessel. So bottom line, plasticity in this case is happening in many different settings, and that's probably one of the reasons why process expression is constantly maintained. Now we started to dissect a little bit more these initial steps, which I told you, which is the specification of lymphatics and then the budding of lymphatics. How does it happen? And this budding, we were very curious because we know these progenitors do not proliferate much into the vein. Therefore, if these cells are constantly exiting the vein, at some point, the integrity of the vein may be compromised and disintegrate, and that will be tragic, as you can imagine. So we did quite a bit of electron microscopy and imaging to really understand how this process happened. And what we were able to demonstrate, Jing Yang, another postdoc in my lab, that this is an active budding mechanism in which the cell additions in the vein do not disintegrate. Instead, what you have is that the addition junctions maintain the integrity of the vein very strong, and these cells start coming out in a very precise manner, and one pulling another one, and they are together by the expression of very strong expression of VE cadherin, as you can see it here in this image here on the side. But how is this budding organized? What makes these cells leave the vein and migrate in a very precise manner? This is not a random budding, it's very polarized. And there was a paper published by this Finnish group I told you before, Kari Alitalo, a few years later, that showed this ligand, VEGF-C, which we know is expressed in the surrounding mesenchyme, is the key player that makes these cells bud off from the vein and eventually move into the mesenchyme of the embryonic, of the embryo, developing embryo, and all these lymphatic endothelial cells eventually form the lymph sacs and eventually form the entire network. And in these mutant embryos, instead, what happened is the lymphatic endothelial cells remain all trapped inside the cardinal vein. So the VEGF-C, as I told you, the receptor is VEGFR3, which is strongly expressed in the lymphatic progenitors, which are inside the cardinal vein. Later on, Satish, again in my lab, was able to show that indeed this process of budding is tightly regulated by the feedback loop between VEGFR3, this receptor, and PROX1. These two genes control each other. We show it by many ways, and this is all published. And that's somehow the mechanism by which the fate of the cells, the identity of the lymphatic progenitors, and the number of progenitors is regulated. So basically, as you can see in this diagram, these cells are leaving the vein, but it's basically through this feedback loop between these two genes that this process is regulated. One thing we never, and this is this paper, so this PROX VEGFR3 feedback loop maintains the identity and regulates the number of lymphatic endothelial cell progenitors. But the problem we didn't know is how this budding stops. When does the system knows that you have enough progenitors and you should stop budding, otherwise you will disintegrate the cardinal vein. And this is the initial process I just was telling you. These are the key players involved in that process: PROX1, the VEGF-3 receptor, and the ligand VEGF-C, and the migration. But and again, you see it here in a better diagram. Again, PROX start to be expressed in the cardinal vein. Then through VEGF-C expression, they leave the vein and then form the lymphatic sacs and eventually the entire network. But how is that the system knows when to stop producing progenitors? And this result came, in fact, last year, and a little bit as an accident, because a colleague of mine at Northwestern published this paper in which he showed that a subunit of mitochondria, complex 3 in this case, is necessary for blood endothelial cell proliferation during angiogenesis. And I must say, my knowledge about metabolism, my mitochondria at that time was very limited, and all what I knew was from high school, the Krebs cycle. And I was very surprised by this paper because I always thought if you don't have mitochondria, the cell will die. Well, it turns out that this was not the case in here. They basically was required for a very specific process. So we decided to collaborate with Nav Chandel, and he provided us with this mouse strain. And because we were curious what happens in lymphatics, we know obviously most cells needs metabolism to fulfill a number of their functional roles. So, and that's exactly what we did. We deleted mitochondria complex 3 from lymphatic endothelial cells, and the result was quite striking. Because what happened in those embryos, and just to remind you, this is complex 3, basically the entire respiration machine is defective. In those embryos, the results were striking because basically these embryos were also devoid of lymphatic endothelial cells everywhere in the body, and this is a conditional mutant. And what we were able to show is that the reason why there are no lymphatics, and maybe this is a little bit complicated, is because normally, as you see in this panel A, the cells the lymphatic endothelial cells, but leave the cardinal vein and they express PROX1 and VEGFR3. However, in the mutants, the expression of VEGFR3 is highly downregulated very early on. You have a few progenitors, but later on these progenitors disappear because as you don't have VEGFR3, this feedback loop I told you between PROX and VEGFR3 is broken. Therefore, the identity of lymphatic endothelial cell progenitor is lost, and you end without lymphatic endothelial cells. So we show that this is through epigenetic modification, basically methylation and acetylation in the locus of VEGFR3. And that's what mitochondria is doing when you don't have it. Basically, VEGFR3 is not active anymore, and therefore you lose the lymphatic fate. So what we think we know now that basically is mitochondria the one that sends the metabolic requirements and said eventually to the system, don't produce more lymphatic progenitors. And this is the diagram of that. This is more or less the summary of what I just told you, of what we know today. And this is, again, the schematic diagram of how lymphatics are formed in the embryonic tissues, more or less what we know today. Through this work and the work of many other labs, one thing that besides identifying how lymphatic formation happens, many genes which are mutated in different forms of lymphatic malformations, lymphedema, things like that were identified. That was very valuable. And now there are easier ways to diagnose these diseases. But the most important thing probably that happened besides this in the last 5 years, I would say, is that our understanding of the role of lymphatics in health and disease has changed completely. This, what you see here, is what is basically in every textbook, medical textbook about lymphatics until a few years ago. Basically, it was considered a boring system that drains lymph fluid from extracellular spaces, absorbs lipids from the intestinal tract, maintains the fluid homeostasis, transport white blood cells and antigens-presenting cells to lymphoid organs. In disease, it's known that basically could lead to lymphedema when genetic or acquired mutation, for example, in breast cancer, removal of lymph nodes produce edema in the region of the arm. But obviously, they are well known in the role in the transport of tumor cells in cancer. And this is more or less what I just told you. This is a summary of the classic roles of lymphatics in lymphedema, meaning the swelling of extremities, in the transport of metastatic cells, and in immunity and inflammation. But our vision of this has changed completely in the last 5 years, and now suddenly lymphatics do everything, pretty much, in whatever functional role you want to think in health and disease. We know now lymphatics are involved, and this is a very exciting time to work in lymphatics. And this is more or less another cartoon of what I just told you. And this is partially, we started another postdoc in my lab that showed that lymphatics are also very important in obesity. If lymphatics are leaky, they promote obesity. And now we know lymphatics may be involved in Crohn's disease, in glaucoma, in cardiovascular disease, in neurological disorders, including Alzheimer's, in many more, which I'm going to tell you in a minute. Regarding the obesity, the way this happened is because we found that our PROX heterozygous mice, they do have lymphatics, but those lymphatics are very abnormal. So abnormal that the majority of the heterozygous animals die at birth with these major issues, which is leakage of chyle into the intestinal tract or in the thoracic cavity, such as the pups die at birth. However, those that survive to adulthood, they become extremely obese. You see a huge accumulation of fat in the visceral part of the animal. And what we showed was that in these animals, the lymphatics are broken, and therefore all the lipid particles, chylomicrons, that should be inside the lymphatics leak out. And what Natasha Harvey, the postdoc in my lab working on this, was able to show that chylomicrons are adipogenic in vitro. If you put it in culture with pre-adipocytes, they differentiate into adipocytes. So what we proposed at that time, for the first time, is that lymphatics can make you fat. In fact, if your lymphatics are defective, they release chylomicrons that contain free fatty acids, and that promote the novo adipogenesis, and then you can become fat. And now some of this data has been validated by other groups in other animal models, as you can see in this recent paper. But now, as I said, lymphatics have been found in other roles. For example, there is a specialized lymphatic on the back of our eye called the Schlemm canal, which is known to be responsible for glaucoma. In the last few years, there's been a lot of excitement because we rediscovered that there is a lymphatic vessel on top of the meninges called meningeal lymphatics, which are very important probably in a variety of neurological disorders, as I mentioned before, in aging, Parkinson, MS, you name it. And these are some of the papers related to that. Recently as well in Crohn's disease, in hair follicles, in regeneration, for example. More recently, 3 papers in cell stem cells saying that they are required for the maintenance of the stem cells that are nearby the intestinal cell niche in the gut. So again, lymphatics are coming with more and more function every moment. And the one I'm going to be talking for the rest of this time, which is the role of lymphatics in cardiovascular disease. And basically, it has been shown that lymphangiogenesis, meaning the growth of lymphatics, improve cardiac function after an infarct. And these are some of the papers showing that when you have an infarct, there is de novo lymphangiogenesis, and somehow that helps to repair the injured heart. So the formation of lymphatics in the heart start in the mouse start around mid-gestation when they start to enter into the heart around E14.5. And what I'm going to tell you is a story we published 2 years ago. Another postdoc in my lab, Shirley Liu, which now she has her own lab, in which what she started to do was to try to figure out what happened if you remove prox conditionally during mid-gestation from the heart at the time that the hearts are entering into, the time the lymphatics are entering into the heart. She induced at E13 and 14 and open at 17.5. And what she saw was indeed these animals, the heart was completely devoid of lymphatics, in contrary to the control that you see all the lymphatics in the heart. But the surprising piece of data was that the heart was 1/3 smaller. And this was really surprising because with that size of heart, you die. And why was it smaller? What she was able to show this was because defective cardiomyocyte proliferation and increase in cell death of the cardiomyocyte, that was the reason why this heart was smaller. But this suggested that the lymphatics were playing an additional novel role, which is be required for organ growth, but also suggested that somehow lymphatic endothelial cells secrete some paracrine factors which are required for cardiomyocyte proliferation and homeostasis. Charlet did a lot of work culturing lymphatic endothelial cells and then using the media to put it in contact with iPS-derived cardiomyocytes, so primary cardiomyocytes, and she was able to show that indeed they are sufficient to promote cardiomyocyte homeostasis. That suggested that, and this she did it also with cultured cardiomyocytes. This suggested again that there was some secreted factor doing that. She did proteomics and mass spec, and we identify a variety of factors we are working at the moment. But the one she selected at the time was an extracellular protein called reelin, which is well known in the neuronal field because mice mutant for reelin die early on from ataxia. Reelin is very strongly expressed in neurons, but it was a report that reelin was also expressed in lymphatics, and again, these are the papers showing that reelin is really important in the CNS. But it was this paper showing that lymphatic was also reelin was also expressed in lymphatics, and Charlet was able to validate and show that indeed in the heart, reelin is very nicely expressed in the lymphatics in the heart and is co-expressed with another lymphatic marker called LIVE1. So the obvious experiment was to see what we saw in those mutants happens also with reelin. We obtained reelin conditional mutant mice and we deleted using the same approach at mid-gestation. And this was very exciting result because she was able to show that the reeling condition of mutant hearts were also 1/3 smaller, consequence of defective proliferation and increased cell death. That was very rewarding piece of data because it confirmed that reeling was the major player involved in that role. And what this indicated is what is in this summary: cardio and lymphatics are required for embryonic heart growth, are also required for cardiomyocyte proliferation and survival, and reeling is the first identified lymphoangiogram factor that regulates cardiomyocyte proliferation and survival, and we identified that integrin beta 1 is the receptor expressing cardiomyocytes. But the other interesting aspect of reeling was when she looked at the expression profile of reeling, she looked, as I told you, it's nicely expressed at D17.5 in cardiac lymphatics. It remains at P2, but it starts to be very strongly downregulated at P7, and you cannot see it anymore at P14. Also, when she did an infarct in neonates to test what happened with these animals, what she was able to see is that reeling is reinduced in the infarcted area, as you can see in this panel very nicely, but not in the non-infarcted area. So, and this was very exciting for a number of reasons, but particularly because, zebrafish heart regenerate, but in mammals, in the case of it doesn't, except in the mouse, the heart can regenerate up to P7. And that was very exciting because that matches precisely the expression profile of reeling, which I told you is expressed more or less up to P7. So what Shaolei did immediately was to basically use reeling mutant embryos and mutant mice and do an infarct, and then see what happened, whether reeling was required for the proper repair and cardiac function of the injury heart. And she was able to show that indeed this was the case. You see in this graph, we measure cardiac function by ejection fraction, but also for the size of the fibrotic area. And as you can see here, cardiac function was really impaired. The size of the fibrotic area was very large. And the reason why this, partially the reason for this was less proliferation and more cell death of cardiomyocyte. So demonstrated that reeling was required for neonatal cardiac regeneration after MI. And finally, what she decided to do is what happened in the adult. I told you that when you have more lymphatics, has been shown that they improve cardiac function. So what she decided to see whether reeling could be a therapeutic approach. And what she did was to do an MI, an infarct in adult mice, wild-type mice, but add reeling in collagen patches and see if that will improve cardiac function. And that was indeed very nicely the case. As you can see in these graphs, you see the animals that have the patches, reeling patches, cardiac function is much more improved compared with the controls, but also the size of the fibrotic area is much more reduced in these animals. So in summary of this part, reeling is required for neonatal cardiac regeneration after MI by improving cardiomyocyte proliferation and survival, and improves adult cardiac repair and function by reducing cardiomyocyte cell death and improving cardiac function after myocardial infarction. So in summary, and I hope you got an idea now what lymphatics are doing much more than we knew before. It's a lot of novel function for lymphatics and many more to be discovered. And I just told you about the novel role in organ growth and repair. And now in the lab, we are also analyzing, we have data that this is not exclusive for the heart, but it's also true for other organs. And we are working particularly in the liver and the lung. And this is true for other factors from our proteomic analysis. We have many other candidates we are validating at the moment to see whether they are responsible also in health and disease for the repair and regeneration of different organs in the mammalian embryo. So this is, in summary, what I wanted to tell you. Basically, a lot of people to thank that collaborated, members of my lab, many people around the world, and obviously the funding from NIH that allow us to do these experiments. And you're welcome to visit us in Chicago. And I hope, sorry that was a little bit in a rush, but if you have any questions, I will be more than happy to answer what you have. Thank you.
¿Hay alguna pregunta aquí en el auditorio? A ver, pasar a Luis.
Hola, Guillermo, Luis.
Hola, Luis. No te veo, pero te oigo.
Okay. Te pregunto sobre la rilina, lo de la última parte. O sea, ¿sabes o has considerado cuál es el receptor de la rilina en el músculo cardíaco?
Sí, eso está en el paper que publicamos. El más importante es integrina beta 1. Y incluso la manera que lo descubrimos fue porque se había publicado un paper hace unos 10 años donde muestra que los mutantes de integrina beta 1 tienen el corazón más chico, igual que los nuestros. Hicimos los experimentos, los dobles heterocigotos, y tienen el corazón más chico también.
Entonces, los receptores, porque rilina en el sistema nervioso tiene otros receptores.
Sí, completamente. Al principio, con los que hablaba, los expertos en relina decían que no podía ser, pero claramente es integrin beta 1. Ahora sabemos que hay un par más de receptores en cardiomyocytes, pero es integrin alpha 1A and alpha A12, I think, or something. Pero no los clásicos de las neuronas, no.
Hola, Guillermo. Una pregunta acá. Durante la regeneración del corazón, se ha visto que justamente la plasticidad que tienen los vasos linfáticos es importante, o es simplemente requerida su presencia.
No, en el caso del corazón, es la plasticidad, y hasta donde sabemos no es muy importante, es la presencia, ¿no? Y entonces lo que estamos ahora tratando de discriminar con las herramientas que tenemos y los modelos de ratón es cuánto influye la relina per se versus cuánto la función clásica de los linfáticos, que es traer células inmunes, remover debris. Entonces lo que estamos haciendo ahora es promover crecimiento de linfáticos en los ratones mutantes de relina y ver cómo funciona la actividad cardíaca en esos animales. Además, generamos un ratón nuevo que ahora expresa relina y la podemos está flux, podemos expresarlo en una tissue and time specific manner.
Una pregunta, ¿existe alguna condición en humanos que se sepa que se debe a una disminución o un defecto en la expresión de PROX1, ya sea metabólica?
No. PROX1, yo digo siempre, todo el mundo me pregunta lo mismo, yo estoy convencido que si tuvieras algún problema en PROX1 sería letal. PROX expresa muchas células, muchos tejidos y es tremenda importante para todos ellos. En cambio, relina sí, a mutantes en relina tienen linfedema, de hecho, los pacientes.
¿Y la opción de usar como blanco terapéutico o algo así sería PROX o sería más como
PROX definitivamente no. Relina es una de las opciones que sí, pero relina es una proteína demasiado grande, es 30 kilobases. Entonces lo que estamos haciendo también es un screening, a chemical screen, to identify small peptides or chemical compounds that could mimic the activity of reelin and could be used for therapeutic approach. But we are really far from there. We need to go to larger animals. It's still a long way to go, but it's very promising.
¿Se expresa todo el tiempo PROX1 en la vida adulta o nada más?
Sí, no, PROX1 está, por eso decía, es necesario para mantener la identidad. Si tú lo quitas, pierden la identidad.
Y es un homeobox gene que tiene una serie de blancos. ¿Cuáles serían los más importantes blancos?
Es muy poco que se sabe sobre eso. Te diría lo más, el que sabemos es el que les dije, VEGFR3, es un blanco de PROX. Y sabemos algunos que están arriba de PROX muy temprano. GATA2 está ahí, CUPTF2 está ahí, SOX18 está ahí, VEGFR3. PROX se autorregula también. Pero hoy por hoy tampoco sabemos demasiado de PROX. Digo, yo en esta conferencia que he estado creo que no hay una sola charla que haya dado que no se nombre a PROX, ¿no? Digo, es una herramienta fundamental como marcador, como anticuerpo, como animales reporteros. Es súper útil, pero para la parte terapéutica PROX no sirve para nada, ¿no? O sea, transcription factors are not therapeutically valuable.
Una pregunta desde el YouTube. Si la linfangiogénesis es similar en el pez cebra que como en mamíferos.
De hecho, es bastante similar. Hay obviamente algunas diferencias puntuales, pero también se ha demostrado muy claramente que empieza en lo que se llama la paracaudal vein y empiezan a salir sprouts que eventualmente se juntan y forman lo que es el lymphatic vessel. También son venous derived. Muchos de los genes in pathway son absolutamente igual. La única diferencia es que parte de los linfáticos de la cara tendría un origen distinto en el pescado, pero básicamente es un proceso muy parecido.
Y la otra parte que mencionabas de la epigenética, del factor de mitocondria,
Ajá.
¿qué hace en la región regulatoria de PROX?
No de PROX, esto es en el otro, en VEGFR3, en el receptor se pega y lo que hace es, activa por metilación el promotor de VEGFR3. Entonces, cuando no está...
Es como un factor muy constitutivo, ¿no? O sea, esperarías que haga cosas.
No es constitutivo. De hecho, VEGFR3 se prende y se apaga.
No, me refiero al de la mitocondria. O sea, supongo que tiene efectos generales en regular varios genes.
Bueno, sin duda, sí, mitocondria complejo 3 hace un montón de cosas, pero en este caso específico, por lo menos la razón por la que no hay linfáticos es porque regula concretamente a VEGFR3, que se prenda a VEGFR3. Entonces, cuando no está este complejo, VEGFR3 se apaga, no puede mantener su expresión y no puede mantener a PROX, y entonces no hay linfáticos. Pero nosotros lo que postulamos que es un sistema muy práctico que básicamente el microenvironment, a medida que las células migran, va determinando el estado metabólico y le dice a las células o al sistema que se prenda o que se apague, ¿no?
Pues si no hay más preguntas aquí, parece que tampoco en línea.
Si alguien tiene alguna pregunta, me puede escribir. Contento de contestarles.
Francisco,
otra pregunta justamente sobre el efecto de las mitocondrias. A mí me llama la atención si es más bien cómo sucede la metilación específica del VEGF mediante el complejo 3. Si se sabe si más bien esto es una consecuencia de que no empiecen a agarrar la identidad de linfáticos o al revés, ¿qué es lo que está previniendo que la agarren desde un inicio?
No, inicialmente la tienen porque, no sé, la pasé muy rápido, pero VEGFR3 se expresa al principio. Lo que pasa se va apagando, entonces un día después ya casi no lo ves. Entonces lo que va ocurriendo es que la activación del locus de VEGFR3 se va cerrando, se va apagando por falta del complejo 3, que es lo que mantiene, lo que te digo, la HK4 methylation and ACT7 acetylation. Entonces VEGFR3 se apaga y entonces pierden la identidad linfática.
Oye, y esto que decías de cuando no está PROX1 se producen más venas, entonces hay un fenotipo también como de exceso de venas o algo así.
Bueno, es difícil decirlo, ¿no? Porque en el embrión básicamente estamos hablando de 14, en el embrión no hay linfáticos y no es que pierdan la identidad. Lo que lo que me refería son estos casos muy concretos que no digo, cuando lo deletamos al condicional, lo que ves es que muchos de los linfáticos empiezan a parecerse a venas y aparecen llenos de sangre. Y después tienen los casos concretos que te dije, como podría ser el caso del tumor o el caso del pez cebra, ¿no? Donde cuando se corta la vena. Pero no es que haya, por lo menos no hay muchos ejemplos donde puedas investigar esto concretamente, que haya más venas, ¿no?
Bueno, pues entonces le agradecemos
Yo quiero preguntar.
Perdón. Hay otra pregunta que no había visto.
Sí, hola, Guillermo, soy Marta Vázquez.
Oh, hola Marta, qué gusto.
Sí, ¿cómo estás? Qué gusto verte.
Igualmente.
Oye, me da curiosidad. ¿Son iguales los vasos linfáticos del cuello que los que están cerca de las patas?
Bueno, los linfáticos son en general todos, tenemos dos tipos básicos de linfáticos, lo que yo mencionaba, los capilares y los más grandes que son los colectores. Y son básicamente iguales en todo, a lo largo de todo nuestro cuerpo. Lo que varía en los órganos, digamos, empiezan a tomar identidades un poco específicas. Por ejemplo, los linfáticos del riñón eventualmente son distintos a los del hígado, por decirte algo, porque, por ejemplo, las adhesiones celulares son distintas en esos dos órganos porque tienen que dejar pasar más fluidos o menos fluidos o están más expuestos a oxígeno o cosas por el estilo. Pero los vasos, los linfáticos son básicamente los mismos.
¿Y la manera como se originan es igual en todos los casos?
Bueno, eso es lo que también hay un poco de debate. Hay algunos que te dicen, por lo menos está el caso concreto del corazón. Hay evidencias de que no todos son derivados de la vena. Algunos se derivarían del lateral plane mesoderm. En el intestino hay alguna sugerencia de que un porcentaje bajo podría tener un origen hematopoyético, pero yo tengo mis dudas de eso. Y hay algunos que argumentan que en algunos órganos habría un porcentaje, no sé, 10, 20% que tendría otro origen, pero no hay ninguna demostración clara y concreta. El único que para mí es convincente es el corazón, que ya te digo, sale de una población islet 1 positiva que del lateral plane mesoderm.
Y mi otra pregunta es en cuestión de ¿cuántos enhancers se sabe que tiene Prox1?
Ni la menor idea. Muchísimo.
Porque tú decías que está regulado por Notch.
Pero, no dije eso.
¿No?
No. Notch regula la identidad arterial, pero no. Hasta donde yo sepa, podría ser, pero no. No tengo evidencias que esté regulado. Pero cáncer de Prox debe haber cientos. Imagínate cuántos se expresan casi todos los tipos celulares. Solo, por lo menos en zebrafish, solo para los linfáticos se han identificado por lo menos 20.
Ok, está muy interesante. Te mando un beso y un saludo a Betty.
Gracias, igualmente. Se cuidan y ojalá
nos
ver en persona.
Ya ven a visitarnos.
Pues Betty acaba de regresar el otro día.
Ya ven a visitarnos.
Yo encantado en cuanto pueda.
Pues una muy bonita plática, muchísimas gracias. Gracias.
Me voy a empacar ahí a dormir. Chao.
Nos vemos, Guillermo.
Gracias.